So hello there and welcome. Today I'm gonna talk about blue-white screening. I think you'll find this video particularly comprehensive because I am going to go into more detail into the molecular mechanisms that form the basis of this technique. But what is this technique exactly, and what is it based on? So blue-white screening is a rapid technique to identify mutants, which are basically plasmids with a gene you have inserted. So it's a selection technique. The whole technique is based on a particular enzyme, which is called beta-galactosidase or lactase. Under certain conditions, E. coli synthesizes, so makes up this protein, which actually cuts, or to be more exact, breaks down through hydrolysis a molecule of lactose into its two main constituents, which is one galactose and one glucose. The whole mechanism has at its base the so-called Lac OPERON. But what exactly is the Lac OPERON? E. coli, of course, has genes in its chromosome. One of these genes, it's called the LacZ gene. Of course, as any other gene, it has its own promoter, which is called the Lac promoter, and another sequence which is called the Lac operator. The LacZ gene codes exactly for the protein that stands at the base of this technique. So basically it codes for lactase. E. coli also has another gene which is called LacI, with of course its own promoter here. LacI in turn codes for a protein that is called the LacI repressor. What this repressor actually does is it binds to the Lac operator, inhibiting transcription of the LacZ gene. Think of it this way. E. coli doesn't like to waste proteins, so it doesn't want to make proteins unless it has to, or unless it will, have something to gain from it. So actually, when lactose is not present, the LacI repressor binds to the Lac operator and the LacZ gene is not expressed, because if you don't have lactose in the environment, it's no use making a gene that will break up lactose. But once lactose is present, what actually happens is that lactose itself will prevent LacI from binding to the Lac operator. So basically it acts like an inducer for the LacZ gene. Of course, this will allow RNA polymerase to attach to the promoter, and the transcription will begin. So basically the bacteria will make lactase. So this is just a basic schematic of the Lac operon. Just for blue-white screening purposes, the Lac operon is more complicated, and let me just show you exactly base by base what gene it has. Alright, so for those of you that don't know, the program I'm using right now is called Snapgene. I will have a video in the description which explains in more detail how to use this program and how you can download it. But for now, let's get into sequence mode right here, and let me show you the Lac operon. So as I said before, this is the LacI gene, which has its own promoter. So basically, the LacI gene will code for a DNA binding transcriptional repressor. Downstream of the LacI gene, you will find the cap binding site, the Lac promoter for the LacZ gene, and the Lac operator. So again, when lactose is not present in the environment, this Lac repressor will bind to this Lac operator, which will prevent transcription of the LacZ gene from this promoter. When lactose is present, the LacI cannot bind to the Lac operator, and of course RNA polymerase can come and initiate transcription from this promoter of this gene, of the gene for lactase. But because just lactase is not sufficient for E. coli to use lactose as a fuel source, the Lac OPERON also contains two additional genes, which are called the LacI here, and another gene which is called the LacA. Okay, so this has been a short view, as it were, of the Lac OPERON. Let's get back to the presentation. So, lactase cuts lactose into galactose and glucose. But for blue-white screening, they are using another compound, which is called XGAL. GAL stands for galactose. Lactase also hydrolyzes XGAL, which in turn immediately transforms itself into an insoluble blue bright pigment. You could induce lactase with lactose, however, in blue-white screening they used another compound which is called IPTG. Why would you induce with IPTG when you have lactose, you might ask? Well, the answer is pretty simple actually. E. coli utilizes lactose as a fuel source. So basically whenever you induce with lactose, E. coli will consume this lactose. In genetic engineering you don't want that. You need to have a strong induction which will last a long time. So, because E. coli cannot actually metabolize IPTG and is not that toxic for the cell itself, IPTG is preferable because it will remain in the environment and it will have a long-lasting induction. Again, in a normal environment, lactose from the outside gets into the cell. It induces lactase, which in turn degrades the lactose. So basically lactose acts both as a substrate and as an inducer. In genetic engineering, you will use IPTG and XGAL in the environment. The lactase gene will be induced by IPTG, which in turn will degrade XGAL, forming that blue pigment. So basically in genetic engineering you will use XGAL as a substrate and you would use IPTG as an inducer. Again, E. coli produces lactase, which cuts both the lactose and XGAL. In blue-eye screening, scientists had to develop a new system. So basically they developed an E. coli strain. There are many strains of E. coli with the LacZ delta M15 mutation. What is this exactly? Basically this mutation, the gene that contains this mutation, produces a truncated or a non-functional form of lactase, which is called the omega peptide. This mutation, as I said, encodes for beta-galactosidase lacking the amino acids from position 11 to 41, thus preventing the enzyme from cutting properly both the lactose and the XGAL compound. Both are inducible by lactose or IPTG. For blue-eye screening to work, you'll need to have another piece in the puzzle. Basically, they also genetically engineered the plasmid, or a set of plasmids, which contain a LacZ Alpha fragment, or also called the alpha peptide. This is an example here of a vector which is called PAC18. Basically what happens is the alpha peptide together with the omega peptide form the so-called alpha complementarity in blue-white screening. they both combine and they form a fully functional LacZ protein or lactase, which does cut and does hydrolyze both lactose and XGAL. Let me show you in more detail by comparing in Snapgene three sequences. Sequence number 1 will be the LacZ Alpha fragment from Pac18, which encodes for the alpha peptide. The second sequence will be the LacZ gene from a strain with the LacZ delta M15 mutation, which encodes for the omega peptide. And sequence number 3 will be the LacZ gene from E. coli K12, which is a strain that does not have any mutation, so it encodes for the fully functional protein. These are the three sequences I was talking about. Here you will have the LacZ gene, which is the wild type in strain K12. Right below it, you'll have the gene, the LacZ gene, which contains the delta M15 mutation in E. coli neb turbo, and the third sequence right here will be the part from the Pac18 plasmid. So basically, this sequence right here will code for the entire LacZ protein. The sequence below that, with the mutation, will code for a truncated non-functional form of the LacZ protein, but as you can see here, in the Pac18 sequence, this encodes exactly for the alpha peptide. So if you combine the alpha peptide with the omega peptide in E. coli neb turbo, you will basically get a fully functional LacZ gene, as you can see right here. So that was the start codon, and this will take you to the stop codon right here. So the gene, the LacZ gene from the wild type variant, is basically identical to the one in the neb turbo strain, except for that mutation right here, which spans from amino acid 11 to amino acid 41. So from here to here, which is the LacZ alpha delta M15 mutation. Okay, so how exactly does this fit into all of this, and why is this useful? As you can see here, this Pac18 plasmid, or any other plasmid that contains the LacZ alpha fragment for that matter, also has a multiple cloning site here, or an MCS, right at the beginning of the gene. So, because it has that multiple cloning site right there, let's say for example that I would like to insert a gene into the multiple cloning site through your typical, let's say for now, restriction ligation reaction. Of course, you can use blue-white screening for Golden Gate assemblies and MoClo standards and so on, but for the sake of simplicity, let's say I want to introduce a Cas9 gene right here in the multiple cloning site. So it gets expressed, so I can do a CRISPR, do a genome modification in bacteria later on. As you can see here, the LacZ fragment gets split in two by the gene that I inserted. So basically the LacZ alpha fragment is no longer expressing. So the alpha peptide will no longer be made by the bacteria. However, the omega peptide will be expressed by the bacteria because it has the omega peptide gene on its chromosome. But because both omega and alpha are required to generate a fully functional lactase protein, this lactase, this truncated lactase, will not cut either lactose nor XGAL. Cloning is never 100% efficient. So let's say, for example, that you would transform your some DNA from your restriction ligation mix into your E. coli NEP turbo that contain the LacZ delta M15 mutation. If you will select with antibiotic, you will get colonies that do have a plasmid, because of course you selected with antibiotics. However, if you also introduce IPTG and XGAL on your plate, you will have two types of colonies. You'll have bacteria that contain the PAC18 plasmid, which got transformed with the PAC18 plasmid that's left over from your restriction ligation reaction, which will have the fully functional lactase gene, so you will get blue colonies, and you will also get bacteria or colonies with the defective lactase. Of course, they will be white because the lactase is truncated and it's unable to basically degrade the XGAL in the environment. So this is how your typical plate would look. These are all the transformants, but you will get some blue colonies and you will get some white colonies. The blue colonies are without your gene of interest because the LacZ Alpha fragment is expressing. However, the white colonies do contain the gene that you have inserted into those plasmids because, of course, the LacZ Alpha fragment can no longer be transcribed because it was split in two by the gene you have inserted. So you will get white colonies. So these white colonies are the colonies that basically contain the mutation you inserted into that plasmid.